This outer part is cortex and the inner part is medulla. There is cortex and medulla. Nephrons are arranged in both. You have cortical nephrons and medullary nephrons. Almost all the Malpighian bodies are in the cortex. The length of the tubule as such, PCT, Henle's loop, DCT, all of that varies in the cortical nephrons as well as medullary nephrons or juxtamedullary nephrons. You don't say medullary nephrons, juxtamedullary nephrons. What are juxtamedullary nephrons? Juxta means what? Juxtaposition. To juxtapose something means what? Bringing close. Juxta is a reference to being close. Juxtaposition of something to something else is getting it closer to the other. Alright? So juxtamedullary nephrons are closer to the medulla, so their tubules are shorter. Cortical nephrons are away from the medulla, their tubules are longer. Otherwise also, if you observe, the PCT comes in, the Henle's loop comes in, goes deep into the medulla, and the ascending loop moves towards the cortex, while the DCT also is closer to the cortex. So it's a different look, different places that we are looking at these tubules. All right. Blood is going into the nephron. Blood is going into the nephron, of course, in the arterioles, and goes into the capillaries of the glomerulus. Whatever is filtered out from the blood is going inside the nephron. Whatever is being filtered out is going into the nephron, and whatever is being absorbed is getting pushed outside of the nephron. Correct? So if you consider kidney as a complete collection of these nephrons, there are so many nephrons, millions of nephrons in the kidney, and there is space in between these nephrons. Space in between the nephrons. So what do you call this space? Huh? What? Column of Bertini are all collecting structures. From the nephrons you collect them. Columns of Bertini are between these pyramids. Between the pyramids. Even inside the pyramids there is space between the nephrons. No? Pyramids are all nephrons. Between the nephrons you have space. You call the space between two specific structures within the body as interstitium. Interstitial space. In the body also, throughout the body you have interstitial space. The lymphatic network begins in the interstitial space. Correct? Lymphatics or the lymphatic vessels are the drainage vessels that will drain fluid from the interstitial space. Accumulation of fluid in the interstitial space needs to be cleared so that the circulation is restored. So interstitium is in between, space in between. The interstitium of the kidney is the space within the kidney which is between the nephrons. Is that clear? So there are two nephrons. There is space in between the nephron, the interstitial space. So if something is getting out of the nephron, where is it going? The fluid is getting filtered. Blood comes into the arterioles. The afferent arteriole carries the blood. The afferent arteriole is going into the glomerulus. Inside the glomerulus, the afferent arteriole is splitting into glomerular capillaries. Glomerular capillaries are porous. They have podocytes which will make the glomerular capillaries porous. It becomes a mechanical filter. So a lot of fluid is filtered out. And it leaks into the Bowman's capsule. And this is a tube. We have taken a section of this tube. So we are looking at it in 2 dimension. So this fluid goes into the PCT. Then from PCT it goes into the loop of Henle, descending loop, ascending loop, and then you have DCT, which curls around and goes to the collecting ducts. All right. So the fluid is passing here. Whatever is passing here, it goes through all this tube. And eventually when it comes here, it goes into the collecting ducts and all the drainage mechanism is there. Eventually it will go and get collected in the urinary bladder. Finally it goes to the urinary bladder, goes to the pelvis. From the pelvis it gets drained into the ureter and goes to the urinary bladder. So whatever needs to be taken out of this fluid, all that is necessary, almost 99% of whatever has been filtered out will be taken back. 99% of whatever is filtered out is taken back. Where is it going from this fluid? It is flowing through this. Whatever is absorbed is getting out of this tube. Whatever is being absorbed is getting out of the nephron. When it is getting out of the nephron, where is it going into? What is it going into? It is going into the interstitial space. It is going into the interstitial space. So now the challenge is, lot of fluid was filtered out with a lot of essential and waste material. Bulk of it is getting pushed into the interstitial space. What is the logical thing to do now? Where should this go now? Where should the fluid that has come out into the interstitial space, which has your nutrients, electrolytes, and all the essential substances, go? Where should it go now? It should go back into the blood. The drainage from this interstitial space is not from lymphatics. It is through a unique special network of capillaries. It is through special network of capillaries. What capillaries? This efferent arteriole branches out into a network of capillaries around this. So there are two types of capillary networks that you find around the nephrons. Alright, there are two types of capillaries that you find around the nephrons. One is peritubular capillary network. Peri means around, perimeter. Peri means around the tubes. Peritubular capillary network around these capillaries. And the other one is vasa recta. Vasa is blood vessels, blood. And this is around the Henle's loop. Vasa recta is a special capillary network around the Henle's loop and the DCT. Alright. Around the entire tubule, you have the peritubular capillaries. So there are two types of capillaries which have the capacity to absorb these things from the interstitial space. Understand? Now you see, why doesn't fluid get absorbed back into the capillaries in the rest of the body, and the fluid gets absorbed back into the capillaries in the kidney? Fluid doesn't get absorbed back into the capillaries in the rest of the body. It is drained by the lymphatic network, it is drained by the lymphatic vessels and then taken back to the heart via the lymphatic circulation. But here in the kidney, whatever needs to be reabsorbed from that interstitial space, what lymphatics are doing in the rest of the body, these peritubular capillaries are doing. They are pulling the substances back into the capillaries. How? We will come there. All right. Three things we have spoken of right now. In the body of the kidney, when you take the section of a kidney and observe a particular segment, particular part of the kidney, what do you find? You will find tubules, nephrons. You will find different parts of the nephrons, space around the nephrons, and in between the nephrons. What is this space called? Interstitium. And you have specific capillary networks around the nephrons. There are two types of capillaries you find around the nephrons. One is peritubular capillaries and the other one is vasa recta. Peritubular capillaries and vasa recta. Peritubular capillaries will contribute for reabsorption of everything that needs to be reabsorbed into the bloodstream. Vasa recta is the one which is contributing for maintaining the osmotic gradient around the loop of Henle and the DCT for concentrating urine. So vasa recta is a part of the counter current multiplier system. Vasa recta is a part of the counter current multiplier system. Peritubular capillaries is responsible for reabsorption. Alright? Keep it this much so far. Is this clear so far? Right. Now, why do we need what is counter current multiplier system? All of that we will see. But concentration of urine has some challenges. Or we need to look at a few aspects of the functioning of the nephron to understand concentration of urine, how urine gets concentrated. You tell me the nature of fluid, whether it is dilute or concentrated. Let us not worry about osmolality, osmotic pressure, gradient, none of that. We will look at it in terms of whether something is dilute or concentrated. Okay? In relation to what? Diluted or concentrated should always be in reference to something, no? In comparison to something. Alright? We will consider that in comparison to the ultrafiltrate. Ultrafiltrate from the starting point. Alright. Now, this is here. The fluid will be in the Bowman's capsule. When the fluid is filtered out, the fluid will be very dilute. The fluid will be very dilute. I will not show the convolutions. I will keep it straight, just for the sake of simplicity. Now, what happens in the PCT? What happens in the PCT? In the PCT, there is reabsorption of predominantly all the nutrients, all nutrients in the form of carbohydrates, amino acids, fatty acids, glycerol, all the nutrients are removed predominantly in the PCT. And also some electrolytes are also removed. Water reabsorption also happens. Everything can be reabsorbed here. Water reabsorption is not the predominant thing that is happening here, but the most important thing that is happening here is reabsorption of all the nutrients and electrolytes, and good amount of electrolytes. All the nutrients, a little bit of electrolytes. So I can say solutes are reabsorbed. Solutes are reabsorbed. Now, tell me what happens to the fluid inside this now. Compared to what it was earlier, this fluid will become dilute or concentrated. This fluid will become dilute or concentrated. Concentrated, huh? It becomes more dilute. When you add solute into a fluid, it gets concentrated. When you remove solutes from a fluid, it becomes diluted. So this fluid, as it comes down in the PCT, it becomes more diluted. More diluted. Now, as you go down the loop of Henle, descending loop of Henle, logically, what do you want to do now? Logically, what do you want to do now? Do you want to take back more solutes or you want to take back solvent, water? It is very dilute now. If you want to take more solute, you will have to put in more energy. You have to create that gradient for this. You have to increase the solute concentration here. But already if you notice, because there has been a lot of solutes that have been absorbed here, the concentration of solutes in this space, in the interstitial space, is high. Osmolarity is high. The high osmolarity, does it favor water to get out of the tubule or does it prevent water from getting out of the tubule? It favors, because this fluid is concentrated. This fluid is concentrated. So it will favor the absorption of water. It will favor the absorption of water. So there is exit of water from the tubule, or there is entry of water from the tubule into the interstitial space. You understand? Clear? Now, As water comes out of this space, what will happen to the fluid that is flowing down the Henle's loop? It becomes dilute or concentrated? It becomes more concentrated. So by the time it reaches the tip of the Henle's loop, the fluid is concentrated. The fluid is concentrated. Henle's loop has a characteristic. Henle's loop does two things. It can absorb water and electrolytes. If it absorbs one, it will become impermeable to the other, and vice versa. So descending loop of Henle is absorbing what? Descending loop of Henle is absorbing water. So descending loop of Henle is impermeable to electrolytes. Opposite is true of the ascending loop. So what will the ascending loop do? The ascending loop of Henle will absorb electrolytes. It will take out electrolytes actively. It will take out electrolytes actively, making the fluid within the ascending loop diluted than it was in the tip of the descending loop, in the tip of the Henle's loop. So here fluid becomes diluted as it reaches the DCT. So in the DCT, what should happen? In the DCT, there should be further concentration of urine. To further concentrate it, what should you do? You should take out water. You should take out water. That further concentrates the urine. Alright. Is this clear? So this is what is happening in relation to the interstitium. What is moving out from the tube, in what segment of the tube, and what is happening to the interstitium is what we are seeing. Alright. Now, what is the role of vasa recta? Vasa recta is very crucial. The flow of fluid in the vasa recta, the flow of blood in the vasa recta is opposite of the flow of filtrate in the nephron. It is opposite to the flow of filtrate in the nephron. Alright. Here, vasa recta that is around this will take electrolytes, countercurrent. Here it is absorbing water, but vasa recta is taking away electrolytes. And as it moves towards the DCT, it will put electrolytes into this space. Why? You are absorbing water. Here. You are absorbing water, making the interstitial space more diluted. You are absorbing water from this space that makes the interstitial space more diluted. If the interstitial space is more diluted, the osmotic gradient will reduce. The osmotic gradient for the reabsorption of water will reduce. So what does vasa recta do? Vasa recta will pull away electrolytes and push it into this space. So increasing the concentration gradient here. So vasa recta that is moving from the descending loop, it will pick up electrolytes from this space, which has already been absorbed here. The PCT is absorbing electrolytes also. The PCT is pulling away electrolytes also, which is tending to increase the osmotic gradient of this space. Alright, and you observe here, the fluid, the volume of fluid that is coming down the descending loop of Henle is higher. The volume of fluid is higher compared to the volume of fluid that is reaching the DCT. Alright, to pull out electrolytes or to concentrate a lower volume fluid is much difficult than to concentrate a higher volume fluid. Understand? So, the gradients are very important. The role of vasa recta is to ensure that the osmotic gradient is maintained. Clear? So, when it is moving around the descending loop, it is picking up electrolytes and it is offloading electrolytes around the DCT, thereby maintaining the osmotic gradient for the movement of electrolytes and water from the nephron. This is the counter current mechanism. Okay, counter current multiplier. Multiplier means what? Alteration of the gradients and managing, changing the osmotic gradients, osmotic pressures. Is that clear? Specific details, now you open your book. Give me one to look into that. Both of you look into the same. You look at figure 16.6. Look at the nephron. Look at the nephron. As the fluid is coming down the PCT, proximal convoluted tubule, what is the osmolarity? 300 milliosmoles. As it is going down the descending loop of Henle, what is happening to the osmolarity? Osmolarity is increasing. Why is it increasing? Because of the absorption of water. Look at the tip of the loop of Henle. The osmolarity is 1200 milliosmoles, which is the highest there, because a lot of water has been reabsorbed. As it moves up the ascending loop of Henle and the distal tubules, so sodium and chloride and electrolytes are being reabsorbed, increasing the gradient, decreasing the osmolarity within the tubule. So here in the DCT and the collecting ducts, you have reabsorption of water. The reabsorption of water from the DCT requires ADH, antidiuretic hormone. Antidiuretic hormone is the one which will keep the aquaporins open. Aquaporins are the channels which will allow movement of water across the membrane. Is that clear? Now if you look at the vasa recta, how is the flow of blood? It is opposite to that of the nephron. So you have to, they have given it like this, parallel to each other. You have to pick the vasa recta and place it on this. Place it on the nephron. You see that? As the blood is flowing down, the vasa recta, that is adjacent to the ascending loop of Henle and the distal convoluted tubule and the ascending loop of Henle, what is happening to the osmolarity of fluid in the vasa recta? It is increasing. Why is it increasing? As it is flowing down, why is it increasing? Why is it increasing? Electrolyte absorption. Electrolyte absorption in the descending loop. No, there is something wrong here. We'll read what they have given. Something is wrong. Mammals have the ability to produce a concentrated with the endless loop and vasa recta also correct. Counter current. See where the in the descending segment of the vasa recta, there is electrolytes being absorbed from the ascending loop of Henle. That electrolytes is picked up by the vasa recta reducing the osmolarity in the interstitial space. The ascending segment of the vasa recta, which is adjacent to the proximal convoluted tubule, is offloading the electrolytes, thereby maintaining the osmotic balance despite the absorption of water. So the descending loop of Henle is absorbing water, which reduces the osmolarity of the interstitial space, which is balanced by the offloading of the electrolytes by vasa recta. The ascending loop of Henle is absorbing electrolytes, which is increasing the osmolarity of the interstitial space, which is rebalanced by the picking up of the electrolytes by the vasa recta. Clear? So vasa recta picks up electrolytes, maintains the electrolyte balance. Vasa recta offloads electrolytes, maintaining the electrolyte balance. Clear? So now we will look at it parallelly. Descending loop of Henle is doing what? Descending loop of Henle is doing what? Absorbing water into the interstitial space. Okay, we'll make it into a table format. There is this. Loop of Henle, vasa recta, interstitial space. Okay. Descending loop of Henle does what? Loads water into the interstitial space. Vasa recta, ascending segment of the vasa recta offloads sodium chloride. Is that clear? Ascending loop of Henle offloads sodium chloride into the interstitial space. Descending segment of the vasa recta picks up NaCl. Clear? Okay. Now, this is the direction of flow. In vasa recta, this is the direction of flow. Counter current is established. Okay. This is descending loop, this is ascending loop, this is descending segment, this is ascending segment of the vasa recta. So this is loop of Henle, this is vasa recta. What is being done? Descending loop of Henle is increasing the volume of solvent in the interstitial space. What will the vasa recta do? Add solvent into the interstitial space so that the osmolarity is maintained. Maintaining the osmolarity is essential for maintaining gradient for flow. Correct? In the ascending loop of Henle, what is the ascending loop doing? Ascending loop is increasing the osmolarity of the interstitial space. To maintain the osmolarity of the interstitial space, what will the descending segment of the vasa recta do? Take away solute or electrolytes from the interstitial space, thereby balancing the osmolarity. That's all. So parallelly throughout, in every segment, in every part of the segment, it is doing that. If the Henle's loop is contributing to increasing the solvent concentration, solvent volume, vasa recta pumps in solute so that the concentration is maintained. If Henle's loop is adding solute, vasa recta is picking up solute. Alright? So countercurrent flow. Where will this vasa recta go and end? The vasa recta goes into the renal vein, capillaries of the renal vein, which will eventually join together to form the renal vein, goes and joins the inferior vena cava. Is this clear? Everybody? Clear? So osmolarity, you should look at it in terms of 300 to 1200 to again some 700, 800. All right. Okay. That's all. Countercurrent. We will read once what is given. That's all. It's only one paragraph. Is it clear, everybody? Take. Any doubts, confusions in this countercurrent mechanism? See how beautifully it is designed. It is designed so well that balance is brought about so precisely. Things that can go wrong here. DCT, distal convoluted tubule, and the collecting ducts also contribute in the absorption of sodium. What is required for the absorption of sodium in the distal convoluted tubule? There is a hormone that is required for the absorption of sodium from the distal convoluted tubule. Which hormone? Sodium absorption, aldosterone. Aldosterone is required. If there is shortage of aldosterone or deficiency of aldosterone, dilute urine can happen because of increased diuresis can happen because of increased sodium concentration in the urine. All the glucose that has been filtered out should be reabsorbed in the PCT. All the glucose that is filtered out should be reabsorbed in the PCT because the further segments are not capable of absorbing glucose. Glucose is absorbed by what? Wherever, whether it is in the intestine or in the nephron, glucose is transported by GLUT, G-L-U-T, glucose transporters. Glucose transporters are co-transport mechanisms. What is co-transport? Co-transport.
Both direction.
No, not both direction. No. In school you will find kids. You also might have been those kids, or you might have had friends of such kind. One child is having urgency to go to the washroom. Bladder is full. But she cannot go without her friend. She's putting, come. Co-transport. Glucose transportation is dependent on the presence of sodium. This glucose transporters have a binding site for sodium. Only when sodium binds to that site, the glucose transporter becomes permeable for glucose. Alright? The significance of this is not much here, but the significance of this is very high in intestine. Intestinal absorption of glucose is dependent on sodium. Alright? So in the absence of sodium, glucose transporters will not be able to absorb glucose. Why is this important? Why is this important to know? Somebody is having diarrhea. Diarrhea is the loss of water and electrolytes. Significant loss of water has happened, significant loss of electrolytes has happened. That means there is deficiency of sodium at the intestinal level also. So you want to make, the person is feeling very tired. You take a packet of Glucon-D, put 2-3 spoons of Glucon-D in water, mix it and give it to the person to give him energy. He is feeling low, so glucose will give him energy. The person will have more diarrhea and he might even collapse. Why? No sodium, so? Why did the diarrhea increase? Why did the diarrhea increase? No sodium, so glucose will not be absorbed. Because glucose is not getting absorbed, it did not contribute to increasing energy. Why more diarrhea? It is called osmotic diarrhea. Glucose is an osmotic substance. Glucose has the tendency to pull water towards itself. An osmotic substance will draw water towards itself. So if you had sodium available, glucose would have been absorbed into the bloodstream and it would have given energy to the person. But because there was no sodium, glucose did not get absorbed, and glucose is an osmotic substance. It will draw more water into the intestines, increasing dehydration or accelerating dehydration and causing more diarrhea. So the best thing to give if you have it is ORS, oral rehydration salts, ORS to be given. It is a proper mixture of all the electrolytes that are required by the body along with glucose also in that. So that will energize. The only thing that needs to be done, or the most important thing that needs to be done when somebody is having diarrhea, is keep giving ORS. Every episode of diarrhea, give 100 ml of ORS or 100-200 ml of ORS. Whatever fluid is lost, you cannot measure it, but just rough 1 glass of ORS for every episode, assuming that at least 1 glass of water has gone. All right. Should not be too precise. And keep observing for the signs. If there are signs of dehydration, give more. If there are no signs of dehydration, the person is well hydrated, keep doing that. 1 sachet of ORS can be reconstituted in 1 liter of water. 1 liter of water. So about 100 to 150 ml every episode of diarrhea. That means 1 sachet of ORS will be sufficient for a person to take care of about 6 to 8 episodes of diarrhea. Alright, it should not be what people do. If they are having diarrhea, they will think that drinking or eating anything will increase the diarrhea and they will not take anything. They go into initial signs of dehydration and suddenly ORS, they'll take 1 liter of ORS and start gulping it all at once. Fluid should not be gulped down. It should be taken sip by sip, slowly, like that only. Keep it closed. You are dozing off, so you might fall on the bottle and you might spill the water all over. Sit straight. She's like, That slow motion nodding when you are sleepy. Okay. All right. This is enough. This is enough for this. Okay. Gene interaction calculations we will do. Supplementary genes, we will take the example of coat color of mice. There are two genes, gene A and gene B. Gene B is responsible for the coat color. But the manifestation of the coat color requires the presence of another gene. So gene A supplements the function of gene B, and helps in the manifestation of gene B. In the absence of gene A, gene B will not be able to manifest. Alright, so this is supplementary, these are supplementary genes. Complementary genes, we will study the example of color of flower in sweet pea. The color is purple or violet color. Purple color is the dominant color, white color is the recessive color. But the presence of, see, there are two genes for this color, for the production of this color. There are two genes that needs to act. Gene A and gene B, let us consider. One gene will produce the pigment and the other gene will produce the enzyme that will convert the precursor of the pigment into the pigment. Alright, like we have proenzymes. Trypsinogen is a proenzyme. Enterogastrin will convert trypsinogen into trypsin. An enzyme will convert the pro form or the precursor form into an active form. So the color producing pigment is in the precursor form produced by one gene. That will be converted into active form by an enzyme which is produced by another gene. If there is only pigment and no enzyme, no character will be produced. If there is only enzyme and no pigment, no manifestation will happen. When both of them are there, there will be manifestation of the color. Alright. So, we will take these examples and just work out the Punnett square to arrive at the phenotypic ratio. Alright. Coat color, gene A is the supplementary gene. Gene B is the pigment producing gene. Coat color of mice. So dominant allele is responsible for producing black color and the recessive allele is responsible for producing brown color. Dominant allele is responsible for producing black color and recessive allele is responsible for producing brown color coat. This is the status of the genes. Alright. In a Mendelian cross, the first generation cross, the parents for the first generation will always be homozygous for both characters on either side. So the first generation will be AABB crossed with small a, small b, small b. Now what will be the code? And if this gene, if this is not available, if this fellow is not available, there will neither be black nor brown, but the color will be albino. When the supplementary gene is not active, the color, the gene that is influenced by that will not get activated, so it will be albino. Is that clear? So there are three color possibilities. There are three color possibilities in the coat of the mice: black, brown, and albino. So what will be this one? This will be black. This will be albino. The first generation. So the second generation will be, all of them will be this. So all of them will be black. All of them will be black. For the second generation, you are crossing two black coat mice. So it is a cross between heterozygous for AB, heterozygous for AB. Now this is the cross you have to work out on a Punnett square. Anything with at least one capital A will give you black or brown based on whether B is capital or recessive, whether there is a dominant allele for B or not. If there is a dominant allele for B, there will be black color. If there is recessive condition in B, there will be brown color. Any condition where there is homozygous recessive for A, you will have an albino. Clear? You do the chart. I will write the A dash B dash is black. What is A dash B dash? This can be either dominant or recessive. This also can be either dominant or recessive. A dash BB will be brown. AA, anything on the B side, will be albino. Is that clear? Whether it is capital B, small b, or small b, if A is homozygous recessive, it will be albino. You can directly write the phenotype in the Punnett square instead of writing the genotypes. Capital A, capital B, capital A, small b, small a, capital B, small a, small b. Black you can write it as capital B, brown you can write it as small b, albino you can write it as A when you are writing the Punnett square. When you are writing the phenotypes in the Punnett square. So this will be black, 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 black. Vale. Are you still getting it wrong? small a, albino, small a albino, small a albino, small a albino. Okay, correct. See, while doing this, instead of writing the genotypes of the Punnett square, what do we have to look for? Look for small a combination. Irrespective of what the B is, you will still get an albino. Small a, small a, small a, small a, these two, small a, small a, small a, small a, these four. You will get albinos. So albino combination done. Rest all will have the supplementary gene functional. Rest all will have the supplementary gene functional. So you will get brown when you have small b combination. Check for small b combination. You have small b combination 1, small b combination 1, and small b combination another one. Rest all is at least one capital A, at least one capital B associated with that. All right. So how many do we have? We have 9 black, 3 brown, and 4 albino. 9 black, 3 brown, and 4 albino. This is supplementary gene ratio. Epistatic gene also functions, recessive epistasis also functions like this. Recessive epistasis also has the same ratio. 9:3:4. If the same condition was dominant epistasis, that means wherever you find A as dominant, the expression will not happen with B. You will have an epistatic character. Then 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12. See, when you look at 9:3:3:1 for a regular dihybrid cross, 9 is dominant, 3 is dominant recessive, 3 is recessive dominant, 4 is recessive, correct? If you look at two characters, 9 is both dominant, 3 is first one dominant, second one recessive, 3 is first one recessive, second one dominant, 4 is one, the last one is both recessive. Now if we look at dominant epistasis, that means if the first gene is dominant, there will be epistatic character. Zero expression, no expression. Then 9:3:3:1, in these four numbers, which are the ones which has the first one dominant? 9 and 3. 9 and 3. So it will be 12. Correct? Clear? The second one, 3 and 1, has the first one recessive. That means the second one will express as per its genotype. So in the third number 3, the second character is dominant. So it will express the dominant character. 1, the second character is recessive. It will express the recessive character. So if it is a dominant epistasis scenario, 9 and 3 will show the epistatic character, no expression. 3 will show the dominant character of the second gene. 1 will show the recessive character of the second gene. So it will be 12:3:1, dominant epistasis. Clear? Recessive epistasis. Recessive epistasis, let's say the first gene will not express, the first gene will be epistatic when it is recessive. 9, both are dominant, so it will express whatever is the character of B will express in 9. The second one, first one is dominant, second one is recessive, so 3 also is there. It will express. In the third one, the first character is recessive, second character is dominant. So we are talking about recessive epistasis. So epistatic character 3. The last one, both are recessive. Our epistatic gene is also in recessive state. So that will also not allow expression. That is also epistatic character. 9:3:4. You see that? I'll erase this. Now we will just use the statements and do the ratios. 9:3:3:1. Okay? We will consider the first gene as A, the second gene as B. We will look at their dominant status or recessive status only. So this 9 represents capital A, capital B. Both have dominant expression. The second one represents capital A, small b. The first one is dominant, the second one is recessive. Third one has small a, capital B. The first one is recessive, second one is dominant. Fourth number represents, both are recessive. Both are recessive. Done? Over? That's all? Correct? So what do we say? Complementary genes. Supplementary genes and recessive epistasis are functioning the same way. When one is present there is expression for the second one. So let us say supplementary gene. Presence of one is essential for the functioning of the other. Is supplementary. Epistasis, presence of one in a particular form is suppressing on the other. It will suppress the other. Supplementary, this fellow will allow expression. This fellow will support expression. In epistasis, this fellow will suppress expression. Ratios might appear same. No problem there. Alright. So let us say supplementary gene A supplements B. If A supplements B, then here again AB expression is seen. Capital A, small b. So here it is. B is expressed. Here small b is expressed. Here no expression. No expression. Correct? No expression, or you can say new expression. New expression. Like black, brown and albino. Albino is not produced by this fellow. Albino is not produced by gene B. But albino is produced because of absence of the expression of this gene. So supplementary gene ratio becomes 9:3:, the new one is 4. Correct? Complementary gene. Complementary gene A and B complements. So for complementary genes is 9:7. Correct? Are you understanding, all of you? You cannot see. It is low, or I should switch off the light? Small. supplementary. A supplements B. So this one, yes, B will be expressed. This one also expression is there, small b will be expressed. This one, new character will be expressed. New character will be expressed. So the ratio is 9:3:4. Complementary, A plus B is required. Expression AB or AB, no expression. So here expression yes, here no, no, no expression. So the ratio is 9:7. 9:7. Dominant epistasis, A, no B. Small a, B or B. Correct? Dominant epistasis means what? When gene A has a dominant allele, B will not express. When gene A has a recessive allele, B will express in its tendency. If B is dominant, dominant character will be expressed. If B is recessive, recessive character will be expressed. So, capital A, capital B, what will happen there? No expression. That is the epistatic character will be there. Capital A, small b, no expression. Small a, capital B will be expressed. Small a, small b will be expressed. So what is the ratio of dominant epistasis? 12:3:1. 12:3:1. 12 will be epistatic character. 3 will be the dominant character of the hypostatic gene. 1 will be the recessive character of the hypostatic gene. Recessive epistasis. Capital A means capital B or small b. Small a means no b. Clear? If it is recessive epistasis, if A has a dominant gene, dominant allele, B will express according to its alleles. If there is dominant allele, dominant character will express. If there is no dominant allele, recessive character will express. If A is in a recessive state, B will not express, there will be epistatic character expressed. So here 9, what will be expressed? Capital B will be expressed because this is recessive epistasis we are talking about. 3, what will be expressed? Small b will be expressed. 3, epistatic character will be expressed. 1, epistatic character will be expressed. So this would be 9:3:4. 9:3:4, similar to the supplementary gene expression. Okay? Clear? So this one is enough for us. This one is enough. If we look at it like dominant, recessive, recessive, dominant, recessive, recessive for both the genes. All right? Done. Clear? And what else we were supposed to discuss? Morgan's wings. Hmm? Okay. Morgan's dihybrid cross experiment is one thing that somebody asked. You asked for it. Morgan's cross. Okay. Morgan's experiments are significant for contradicting or picking up deficiencies in Mendel's dihybrid cross. It is same. The principles of cross is the same. Morgan crossed Drosophila melanogaster. What is it called? It is referred to by some title. Fruit fly is its colloquial name. What is the title of Drosophila? Huh? It is the guinea pig of the insect world. Experimental. Lot of experiments using insects will be done using Drosophila. A lot of animal experiments are done using guinea pigs. So if somebody is subjected to some experimental trials, you will use the reference as he is being made a guinea pig. He's being made a guinea pig. Okay. Why guinea pigs? Small, easy to handle, easy to study, faster reproductive rate, generations change quickly, all of those advantages. They are very cute. Guinea pigs are very cute to handle. Okay. Observations of what Morgan did gave deviations from the Mendel's ratios. He did all the experiments. He selected two characters. What are the two characters that Morgan selected for study in Drosophila? The body color and the wing shape. Body color and the wing shape. Body color, gray and brown. Gray and brown. Gray is the dominant character, brown is the recessive character. Normal wings and vestigial wings. Normal wings and vestigial wings. Vestigial wings are smaller wings. Normal wing is dominant and vestigial wing is recessive. We will do this only. If it was normal Mendelian cross, the ratios should have been, what should be 9? Grey colored flies with normal wing. Grey colored flies with normal wing. This should have been grey colored flies with vestigial wings. This should be brown colored flies with normal wing. Brown colored flies with vestigial wing. But that did not happen. For selection, how would you select? The first generation parents, who do you select? What would be the parental flies? First generation. Grey colored flies with normal wings, homozygous for both characters. The other one would be brown colored flies with vestigial wings, both of them being homozygous for that character. Now when you go to the second generation, this is what you have to see. Okay, we'll write it down. Parents, grey normal wing and brown vestigial wing. Brown vestigial wing. This would be second generation. What should you notice? You should notice this as grey normal wing. This is grey vestigial wing, brown normal wing and brown vestigial wing. These two were lesser in number. Those two were lesser in number. You had more insects, more than 9 with gray normal wing, more than 1 with brown vestigial wing. What does this mean? It means that the independent assortment has not happened as Mendel proposed. Gray tends to go more with normal. When gametes are being formed, the allele for gray color goes along with the allele for normal wing more often. The one with allele for brown color and vestigial wing goes more often together. Independent assortment says that these should not be connected to each other. They should independently assort randomly, giving you all possibilities of combination. But it did not happen. The ratios did not show, the ratios did not support that, in the fact that there were more parental varieties found than the recombinant varieties. So independent assortment has not happened. So this laid the foundation for the study of linkage. Gray and normal wing, the alleles are linked somehow. Brown and vestigial wing alleles are linked somehow. Now we know what is the linkage. How are they linked to each other? They are on the same chromosome. Gray color and normal wing, the genes for color of the body and the wing shape are on the same chromosome, making it more likely for the parental combination to carry forward. That we'll do it with chromosomes. I will use black color for gray and normal wing allele carrying chromosome, and blue color for brown and vestigial wing carrying chromosome, allele carrying chromosome. Okay. The first generation would be this, no? Parental generation was this, correct? Homozygous for both alleles. The first generation would be, all of them would be this. First generation, all of them would be this, correct? One chromosome came from the father, one chromosome came from the mother. The second generation, both of them, in the first generation parents, both of them have this, correct? Now when gametes are being formed in this will form two gametes, this will form two gametes. So what will this have? One will have black chromosome and one will have blue chromosome. This one also will have black chromosome and this one also will have blue chromosome. Correct? Chromosomes are being segregated, not alleles. Chromosomes are being segregated and not alleles. So black chromosome is what? This is gray and normal wing. This is brown and vestigial wing. Brown and vestigial wing. Now, it has to be this or this, this or this, this or this, this or this. That's all possibilities. Those are all the possibilities. Correct? That is all possibilities exist. So this would be black and black. This would be black and blue. This would be black and blue. This would be blue and blue. Correct? Blue and blue. Now we will alter this a little bit. I will make the vestigial wing allele with dotted lines. I mean the allele for wing shape as dotted lines. Correct? Here what do we have? Black with both straight line and both solid line and dotted line coming together. Blue with solid line and dotted line coming together. What has not happened is black solid line and blue dotted line. Blue solid line and black dotted line has not happened. So if we say black solid line and blue dotted line, what would it be? Capital B, small v. Not happened. If we say blue solid line and blue solid line and black dotted line, it would be small b and capital V. That would not be possible if the entire chromosome is getting segregated. So no recombinant characters would be possible if the linkage was absolute. The increase in the number of parental combinations compared to the expected number of recombinant varieties indicated that the alleles were linked to each other. Linkage was established. But if it was linked, why do we at all see recombinant varieties? Why do we see black solid line and blue dotted line also? Some of them, if it was not 9:3:3:1, it might be 11:1:1:1:4. 11:1:1:3. That might be there. But why is that 1:1 also coming? Why is that recombination also happening? That is because of crossing over of segments. Here you see, this is the parental variety. This is the parent for the first generation. You have black dotted line and solid line, blue dotted line and solid line. Now because of crossing over during meiosis, there is a possibility that this segment could have come here, this segment could have gone there. So some of the chromosomes could be like this: black solid line and blue dotted line, or blue solid line and black dotted line. Possible? In that case, recombination also is possible. So this experiment of Morgan is what proved that Mendelian inheritance is not absolute. It is not the way that inheritance happens. Factors are right. Alleles are there. Genes have alleles, all of that is there. But independent assortment does not happen. Independent assortment of factors is not happening. Independent assortment of chromosomes are happening. The alleles which are closer to each other on the chromosome are less likely to segregate. The alleles which are far away from each other on the same chromosome are more likely to segregate. The alleles which are not present on the same chromosome will definitely segregate. Correct? If they are not present on the same chromosome, they are not linked. So independent assortment will apply to those characters which are not represented on the same chromosome. Independent assortment will not apply entirely to the alleles represented on the same chromosome, or the characters represented on the same chromosome. Independent assortment will be more likely, not completely for the same chromosome, linked alleles, linked genes, if the genes are far from each other on the chromosome, because that will increase the possibility of crossing over and recombination. But if the genes are closer to each other on the chromosome, the probability of crossing over and recombination will reduce. So strength of linkage means how close the genes are on the chromosome. How do you relate strength of linkage and recombination? How are they related? Link them mathematically. Inversely related. Correct? The closer the genes are on the chromosome, the lesser will be the recombination. The farther the genes are on the chromosome, more will be recombination. You cannot say recombination will definitely happen. Alright? So this is the significance of Morgan's experiments. All right. So, many gaps filled. We'll take a 10-minute break. And when we come back, we will discuss genetic code. Genetic code, those properties of genetic code is what we have to study. We will read from the book and quickly do it. And after that, whatever is the next topic that you all want, we'll discuss that. All right. Break for 10 minutes. 10:55, we will reassemble at 11:05. Not 500, 450. Huh? What? You are telling me the brand of the chocolate? You are asking me. What did you say?
300, not 450.
If chocolate only, melody score only
300? No. I am confident that some of you will cross 450. I know the difficulty level of the papers that were given in the last 4 or 5 tests. Since the last visit, since November 1st, the papers are of old syllabus and of a little higher difficulty level. All of you have performed well on the regular previous year papers, which is the difficulty level of the NEET. Now, tomorrow's paper will be a little closer to that. Okay. There is a mic fitted in your voice box only. What was supposed to be a very soft whisper turned out to be a loud noise, which even the last benchers could hear. Okay. So let's see tomorrow attempt. All right. We'll keep it that way only for the rest of the papers from now on. We'll keep it true to the difficulty level and pattern. So let's prepare that way only. All right. Okay. One challenge I gave like this to our school children. Every year, every day they do yoga, they play, they are physically fit. Our school children are physically fit. Very aggressive kids. Aggressive in terms of they are active, energetic. They are not dull, lethargic, laid back kids. Okay. Every year during International Yoga Day, 21st June, we will encourage our children to do 108 Surya Namaskaras. 108. You are doing 12, no? 108. Last year, 38 students did it from class 5 to class 9. This year, I assumed because 1 year they have trained and there will be more students who will be doing it, I was prepared with 50 chocolates. 50. Then by the end of it, when I got the count, it was 89 students who did 108 Surya Namaskaras. From class 4 to 10. We have class 4 students also doing 108 Surya Namaskaras at a stretch. And after doing 108 Surya Namaskaras, they went on to play for 1 hour also. So, physical strength and fitness is very important. So, let me see. Chocolates, how many chocolates? Anyways, I saw that there is that fruit jelly available in the shop outside. It will be good chocolate. I'll give a good chocolate. Which one? Ferrero Rocher. Anyways, whatever. Get 450 first. I'll decide the chocolate based on the number of people who cross 450. Right. Genetic code. Or did I get too enthusiastic and say 450? We'll make it 500 only. Page number? During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotides to synthesize a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences, though, to support the notion that change in nucleic acid genetic material were responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. See, we have seen the sequel of this movie. Transcription, translation, we have studied. It is like that movie came first, and then we are looking at the prequel of the movie, which is the genetic code. Nucleic acids are known to be producing the nucleic acids are the code for producing amino acids. It is not that the nucleic acids become amino acids, but it was also studied that change in nucleic acid sequence, change in nucleic acids led to a change in amino acids in the protein chain, in the polypeptide that we find. So it was extrapolated that probably the nucleic acids have a pattern. Probably the nucleic acids have a pattern, and that corresponds to the amino acids. Now we know, we study it as theories, but when nothing is known, when it is not known, you know that nucleic acid is just a sequence of nucleotides. Even that, it's a chemical first. Then you study the repeating units, nucleotides. Then in those repeating units, you identify the differences. Then you say the difference is because of the bases, nitrogenous bases. Then when you do a lot of study, you will find out there are 4 bases which are repeating in various sequences, which are continuously repeating throughout the nucleic acid. So the difference is only that, no? The difference is only these 4 bases, nothing else. Everything else is the same. The 4 bases, the sequence of these bases is what causes the difference. So polypeptides produced by translation, polypeptides produced as a result of translation of the code on the nucleic acids or the information on the nucleic acid is translating into amino acid sequences, that is polypeptides. So natural correlation is there should be something corresponding to each amino acid. The entire polypeptide should have correspondence, should have some complementary sequence on the nucleic acid, on the DNA. They called that as the gene. And in the gene, there should be something that should correlate to each amino acid, which will say which amino acid should come after what. Those are the initial thought processes that led to the, hypothesizing of the presence of a new genetic code and eventually figuring out what it was. That is the difficult part. Now it appears very simple. It is just, there are 4 alphabets. Every 3 alphabet is a code. Based on the sequence of alphabets, you have different amino acids corresponding to the different sequences of alphabets. But the first step, the paradigm shifting innovations or inventions or discoveries are the ones which are the most important. That is why, though Mendel was wrong in many of his conceptualization, he was not completely right. He was partially right. At present, for the current understanding of genetics, you can consider Mendelian genetics as just basics of it, just the fundamentals of it, principles of it. Yet, Mendel is the most important person in the study of modern genetics. That's why he's called father of genetics, because what he did in the circumstances that he lived in and with the body of knowledge that was available for him to base his hypothesis on, Mendel's work becomes the most significant. So whatever you do that will change the way people start thinking, that is most important. What people build on that later, all of that can go. It will go on and on and on. It can change into something very huge. But that first initiation becomes very important. If determining the Biochemical nature of genetic material and the structure of DNA was very exciting. The proposition and deciphering of genetic code were most challenging. It, in a very true sense, it required involvement of scientists from several disciplines: physicists, organic chemists, biochemists, and geneticists. It was George Gamow, a physicist, who argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. Same things that we discussed now. There should be something that combination should happen, and 20 amino acids have identified. And even that 20 amino acids is what constitutes most of our proteins that we know of. There can be something else also, we don't know. But as of today, we know there are 20 amino acids. The code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. Why? Why 3 nucleotides? Why not 2 nucleotides? Why not 4 nucleotides? 4 nucleotides, 2 nucleotides, if we consider it as 2 nucleotides, the code is of 2 nucleotides. What is the possibility of unique codes? 16. There are 4 bases. You have to make combination of 2. It would be 4C2. 4C2, factorial 4 divided by factorial 2 into factorial 2. Correct? Factorial 4 is, no, it is not combination, it is permutation. Permutation. 4 bases are there. The first position can be held by 4, not even 16. It would be factorial 4p2 is factorial 4 divided by factorial 4 minus 2. 8 by 2, 4. No? No, for each position. For each position. How many possibilities are there for the first position? 4 possibilities. Any of the 4 can come in. For the second position, any of the 4 can come in. There is no condition on the first one. If there is one alphabet in the first position, it should not come in the second position. No such things. So for the first position, you have 4 possibilities. For the second position also, you have 4 possibilities. So 4 into 4, 16 possibilities would be there. But there are 20 amino acids. You cannot have the same code for 2 amino acids. So codon being a 2 base codon was ruled out because we knew that there were 20 amino acids. Then what if it is 3 bases? 3 bases form a code. Then there are 64 possibilities. It will definitely be sufficient for coding 20 amino acids. Why not 4? Why not a combination of all 4? Then it would become 256 possibilities. That will still code for 20 amino acids. It was not 2 base code was easy to arrive at, because you need to have at least 20 codes, 20 codons to code for 20 amino acids. Logically that was easy. But why it is 3 only? Why not 4? Why not 5? Why not 6? Anything beyond that? To arrive at 3 was a challenge. How did they do that? They have done it. Congratulations to all of us. Providing proof. See, the next thing is telling that only. Okay. The code should be made up of 3 nucleotides. This was a very bold proposition. Proposition is not a proof, a hypothetical assumption or a hypothetical assertion. You hypothesize something based on logic, and then you assert that this is how it is, and then you initiate the experimentation to collect data to verify your hypothesis. The data when analyzed can validate your hypothesis or negate your hypothesis. Either case you win. That's the beauty of science. Science, as compared to faith, is when we talk about anything from the point of faith, we always want to prove it to be right. Science is not like that. You say codon is made up of three bases. That is your hypothesis. Then you go on to set up experiments, you do experiments, you get data, you analyze data. All of that eventually gave you evidence that codon is made up of 4 bases. Genetic code is 4 bases. Let us say it shows that. Or we'll consider the other way. I started with the hypothesis that it is 4 bases. 4 base codon is one code. Then I conducted experiment and then I find out that it is 3 bases. So my experiments disproved my hypothesis. But I will still be recognized, rewarded, appreciated and celebrated. Because in science, it is not an attempt to prove your assumption or assertion or hypothesis that will give you recognition. Unraveling the truth is what gives you recognition. Unraveling the truth is what gives you recognition. Even if your hypothesis was opposite of that, you will still be appreciated. So you are not going out to prove your point of view. You are going out to find out the truth. The pursuit of truth is science. Again, truth relative to what our understanding can allow us to explore. It's always limited. Absolute truth, nobody talks about. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khorana was instrumental in synthesizing RNA molecules with defined combinations of bases, hypopolymers and copolymers. Marshall Nirenberg, Khorana and Nirenberg were the ones who did this. So, Marshall and Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Okawa enzyme, polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner, enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in table 5.1. Checkerboard is that just combination of bases. You see that table 5.1. You have all the 3 bases represented to mention which codon codes for what amino acid. All right? The salient features of genetic code. The salient features of genetic code are codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acid. Hence they function as stop codons. Stop codons are which one? Ochre, opal, amber. UAA, UAG, UGA. UAA, UGA, UAG. Ochre, opal, amber. That's the name given to them. Which one is ochre? UAA. Opal? UGA. Amber? UAG. Don't bother to remember it so specifically. If the name of the specific stop codon is asked as a question, that would be a very wrong way to be questioning you, so you should not be bothered about answering that. Dump it. Some amino acids are coded by more than one codon, hence the code is degenerate. Degeneracy of triplet codon, degeneracy of genetic code is very, very important. What is the significance of degeneracy? See, there is some functional significance of the genetic codon being degenerate. What is its significance? It is like a backup defense against harmful mutations. A backup defense against mutations. Now, mutation can change a particular base. What is changed if it is still the same amino acid that is coded even after mutation? Same sense mutation. Same sense mutations are at least not causing any harm to you. There is no change in the protein being produced. Same sense mutations have become safer because the change in the nitrogenous base or the mutation does not lead to the change in amino acid, which does not lead to the change of any sequence of polypeptides. Alright, degeneracy is protective. It is not that it has been designed to be protective against mutations, but the fact that genetic code is degenerate, meaning more than one codon codes for some amino acids. One amino acid can be coded by more than one codon. That is a protection against harm that can happen because of mutation. The codon is read in mRNA in a contiguous fashion. There are no punctuations. The codon is continuous. You don't have any gap. It is not like three bases you read and 2 bases you skip. No, it is continuous. What is the direction? It is directional. In what direction do we read codons? Everything in nucleic acids, we look at it from 5 to 3, 5 prime to 3 prime, 5 prime to 3 prime. DNA replication, 5 to 3. mRNA transcription, 5 to 3. Translation, 5 to 3. Everything in 5 to 3 direction. Okay. What is the sense strand is? During transcription, what is the polarity of the sense strand? Sense strand is 3 prime to 5 prime. Huh? Okay. Now there will be some fun. Okay. Sense strand is the template strand. True or false? Sense strand of DNA is the template strand for a transcription. Dun, 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 dun. Now the by default music that goes in my head are those Cocomelon rhymes. Even now the spontaneous music that came was, could somebody recognize? What is that? If you're happy and you know it, clap your hands. Clap, clap is what my daughter says. So, okay. Tell me, what is the template strand? Is it the sense strand or the anti-sense strand which is the template strand? Sense strand is the template. So what is the polarity of the sense strand? See, tell me, what should be the direction of translation? 5 to 3. So the code should be 5 to 3. In which direction should you read the code on the DNA? In which direction should you read the code on the DNA? 3 to 5, huh? Because you want to somehow make the sense strand the template strand. The code also on the DNA is 5 to 3. Everything is 5 to 3, remember it that way. Everything is 5 to 3. The code has to be read from 5 prime to 3 prime direction on the DNA. So that should be the exact copy that you make as an mRNA. Why do you want to call something as a sense strand? Which makes sense of the code. Should you consider the DNA with 5 to 3 polarity or 3 to 5 polarity as the sense strand? 5 to 3, because that is the code. That is the code that we want to read. That is the sense that we want to make. mRNA should be exactly like that. mRNA should have 5 to 3 same sequence. So mRNA should be built complementary to, mRNA can be built as complementary, no? You don't take photocopies, you build complementary copies. You need a photocopy of the sense strand. So can sense strand be the template? Can sense strand be the template? Anti-sense strand should be the template. So that mRNA is built exactly like the sense strand. mRNA is built exactly like the sense strand. Correct? Okay, anyways, we will move ahead. The code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine. Initially, in the earlier days, specific codons were given, and what does it code for? Used to be asked. Now, you have to remember a few codons. Phenylalanine you remember, because phenylketonuria is a mutational disorder. And sickle cell anemia is because of what changing into what? Huh? What? Glutamic acid changing to valine. So glutamic acid codon you should remember. What is the codon for glutamic acid? GAA, GAG. Valine is GUA, GUG. Correct? So glutamic acid, valine, phenylalanine, methionine. Remember these four. Remember these four. That's all. You cannot remember all the codons. You can remember if you put in efforts to remember that. But putting in so much effort to remember the codons and the specific amino acids is not necessary at this stage for NEET. If you get into genetics, then you will have to remember all of that and even their variations. Right now for NEET, it is enough to know only 3 amino acids: glutamic acid, valine, phenylalanine, and methionine. Methionine is AUG. Methionine is AUG. Okay. Okay. Clear? Methionine, phenylalanine, glutamic acid, and valine. That's all. And the 3 stop codons: UAA, UGA, UAG. Who is good with mimicry? Who can do mimicry? Okay. Those stop codons were very confusing for me when I learnt it in 12th standard. But you have to remember, those were the times in our state's entrance exam, they used to ask these codons specifically. We tried to memorize that entire table. All of that failed. Stop codons at least we should remember. I have a friend. He got a hack for that. How to remember. Can somebody cry like a baby? Look at those stop codons. Look at the stop codons. Don't they appear like the script for the baby's cry? UAA. UGA. Right? Huh? UAA, UGA, UAG. Use that as a script for baby's cry. You will never forget stop codons. Okay. UAA sounds like? Okay. DNA is double stranded. 5 prime, 3 prime, 5 prime, 3 prime. 5 prime, 3 prime. Alright? So let us say the gene is here. This is the locus of the gene. This is the locus of the gene. Okay? Let us say the gene is XY. The gene is XY. Complementary strand here would be YX. Complementary strand would be YX. Let us assume that way. What do you want the mRNA to be? The mRNA should be XY from 5 prime to 3 prime. Correct? Yes or no? mRNA should be XY from 5 prime to 3 prime. This segment you want. This segment you want. Can it be? Strand is the template strand. Alright? You find errors in some books, mixing up. Just go by logic. Sense strand is the code that we read on the DNA. It is 5 to 3 and the mRNA should be exactly like that. So mRNA should be 5 to 3, similar to what is there on the sense strand of the DNA. So you have to use the template which is complementary to the sense strand. Clear? Got it? Move on. The code is nearly universal. For example, done. AUG has dual functions. It codes for methionine and it also acts as an initiator codon. Codes for methionine and initiator codon. UAA, UAG, UGA are stop terminator codons. If following is the sequence of nucleotides in mRNA, predict the sequence of amino acid coded by it. Use the checkerboard and tell the sequence of amino acids. Just some fun activity. AUG is methionine, UUU is phenylalanine, UUC is phenylalanine again. Methionine, phenylalanine, 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 phenylalanine. Enter methionine with everything else phenylalanine. Correct, no? That's all. Now try the opposite. Following is the sequence of amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. Same. Do you face any difficulty in predicting the opposite? Is there any difficulty in predicting the opposite? Can you predict the sequence of nucleotides by looking at the sequence of amino acids? Exactly. Why not? Degeneracy. You don't know which codon is actually coding for that amino acid when the amino acid is degenerate. When the amino acid is coded by a degenerate codon. So phenylalanine is there. For the rest of the sequence is phenylalanine in the amino acid sequence. Phenylalanine has two codons. Which one do we consider? Correct? With the sequence of nucleotides, with the sequence of nucleotide bases, you can predict the amino acids perfectly. But reverse is not accurate because you don't know which amino acid is, which base is there in the codon. Okay. Can you now correlate which two properties of genetic code you have learnt? Okay, which two properties of genetic code? Degeneracy and what other property? Okay. Why only two properties? We have read this in continuity. So genetic code is continuous. Then specific. Genetic code is specific. One codon codes for one amino acid only. Not true with the opposite. One amino acid need not be coded by one codon. One amino acid can be coded by more than one codon. So, specificity of genetic code and degeneracy of genetic code are in opposite directions, but not similar. Correct? Specificity of genetic code is code to amino acid. Degeneracy is amino acid to code. They are not similar because code to amino acid is 1-to-1, but amino acid to genetic code is not 1-to-1. It can be 1 to many. Is that clear? Okay. Mutations and genetic code. Do we do that also? Mutations also? With that? Okay, we'll do it off. The relationships between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effects of large deletions and rearrangements in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acids residue glutamine to valine. It results into a disease condition called as sickle cell anemia. Effect of point mutations that inserts or deletes a base in structural gene can be better understood by following simple example. Consider a statement that is made up of following words, each having 3 letters like genetic code. Ram has red cap. If we insert a letter B in between has and red and rearrange the statement, it would read as follows. Ram has Bray DCAP. Becomes nonsense there. Changes. Good example, no? So if you insert 2 letters BI, now would say Ram has BIR EDCAP. 3 letters together say big. The statement would read Ram has big red cap. The same exercise can be repeated by deleting the letters. The conclusion from the above exercise is very obvious. Insertion or deletion of 1 or 2 bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutations. Insertion or deletion of 3 or its multiple bases. Insert or delete in 1 or multiple codon. Hence, 1 or multiple amino acids and reading frame remains unaltered from that point onwards. Important. See, in mutations, you have 2 types of mutations. Point mutations and frame shift mutation. The example that is given here is frame shift. You add codons or you remove codons. The reading frame of the codons will get altered. There will be change from the point of addition or deletion. If there is insertion of codons, what will happen to the frame? Frame shifts away or closer. Frame shifts away, frame gets pushed. Insertion pushes the frame, deletion pulls the frame. Either ways, the sequence gets altered, the sequence of codons get altered, thereby altering the sequence of amino acids. When there is insertion or deletion of 1 or 2 bases, 1 or 2 bases. But if it is multiples of 3, being inserted or deleted, then even there, if it is exactly at multiples of 3 gaps, then there is change at that point and everything else remains the same. If big was inserted in between R and A of Ram, then big was inserted between has and red. So it became Ram has big red cap. But if big was inserted, it's multiple of 3 only, no 3. If 3 codons were inserted in between 1, 2 or 2, 3, then it will still change. So that is a misleading example. Insertion and deletion will alter the frame. The alteration will change the sequence of amino acids if 1 or 2 codons are inserted, if 1 or 2 bases are inserted or deleted. But insertion or deletion of 3 bases may or may not alter the frame except for the inserted or deleted portions. May or may not. It may not alter the frame if it gets inserted or deleted in that chunk where it is not in between a codon. So if 4, 5, 6 is removed, then 1 amino acid will be lost. But 7, 8, 9, 10, 11, 12, all of that will continue to produce the same amino acids, and the sequence of polypeptide that is produced will have all the amino acids minus 1 that is deleted. There will be defective polypeptide, but it will not be an entirely different polypeptide. If 3 codons are inserted, let's say it is inserted between 3 and 4, then again 4, 5, 6 should have produced amino acid X, but it is producing the amino acid which was inserted, which corresponds to the inserted codon, but rest all will remain the same. But if insertion or deletion happens between the triplets, within the triplet, so between 1 and 2 you insert 3 codons, 3 bases, it will alter the entire sequence. So if 1 or 2 bases are inserted or deleted, it will certainly alter the reading frame. But if 3 bases are inserted or deleted, it may or may not alter the reading frame. That should be the statement. We'll relook at the statement whether it is given or not. Insertion or deletion of 3 or its multiple bases. Insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards. Is it correct? It is not correct. So we have to send a letter to Shweta Uppal. Why? No, she is the editor. But where is the content team? They have not mentioned individual names. Oh, they have mentioned. K. Muralidhar, Professor, Department of Zoology, University of Delhi. Boss, there is a mistake in this statement. Please correct it next edition. Okay, done. Okay. Frame shift mutation, point mutation. Point mutations can be same sense, missense, nonsense. Same sense mutation is the altered base gives rise to a new codon which still codes for the same amino acid. That is same sense mutation. Okay. Missense mutation is the altered base gives rise to a new codon which will code for a different amino acid, but it still codes for some amino acid. That is missense. Amino acid gets changed, but it still codes for, there is an amino acid, it doesn't stop. Nonsense mutation is UUA codes for what? Phenylalanine. UUA. Wait. AUG codes for methionine. UAG codes for stop codon. So if there is AUG, instead of A in that chain, U is replaced. A is replaced by U. So AUG becomes UAG. The coding stops there. The translation stops there. That is nonsense mutation. Nonsense mutation is when the codon that is produced as a result of mutation becomes a stop codon. So same sense, missense, nonsense. Frame shift mutation, insertion, deletion. That's all. Okay. That's all. Okay, what next? Cockroach. We'll keep it for the last so that you don't have to revise much. Or we will look at the diagram and try to understand some key aspects of cockroach. Okay, we will understand its salient features. We will do. We will not keep running away from cockroach. There are some specialities of cockroach. Before we go into reading from the book, can you tell me what unique features cockroaches have? They can withstand? Just a moment. Hello? One nimisham. C 8047. 8047. Okay. Sorry. Yes. Tax filing, I had to give an OTP. GST, okay. They can withstand high temperatures, they can withstand nuclear explosions. Why? What gives them that power? Their exoskeleton. And you chop off the head of the cockroach, it can still survive. It will die because of not having food. But it can still keep walking around. Why? Ganglia. They have peripheral ganglia throughout the body. Every segment has a pair of ganglia. So that means their neural regulation, the regulation of their body happens at multiple levels throughout the body. All right? Okay. Their mouth parts, what type? Chewing and biting type. Antenna, very sensitive, chemosensitive antenna. Chemoreceptors are there in the antenna. Chemoreceptors, they have pressure sensors. Eyes, what eyes? Compound eyes. They have compound eyes. What type of vision does compound eyes provide? Mosaic vision. Okay. Give. We'll see what is there in the book and. Why is this book heavy? Old, huh? Old book. There is no cockroach. Then why do we have to study cockroach? Huh? NCERT syllabus. I mean, NEET syllabus. Okay. We'll have to talk to Muralidhar, textbook committee chairman. Cockroaches are brown or black-bodied animals that are included in class Insecta of phylum Arthropoda. Bright yellow, red and green colored cockroaches have also been reported in tropical regions. Their size ranges from quarter inch to 3 inches and have long antenna, legs and flat extension of the upper body wall that conceals head. They are nocturnal omnivores that live in damp places throughout the world. They have become residents of human homes and thus are serious pests and vectors of several diseases. The adults of the common species of cockroach, this is important. The common species that we find in our country is Periplaneta americana. What you need to remember in these things, they are from quarter inch to 3 inches, all of those are insignificant from the need perspective. Periplaneta americana, that species is important. 34 to 53 millimeter long with wings that extend beyond the tip of the abdomen in males. The body of the cockroach is segmented and divisible into 3 distinct regions, head, thorax and abdomen. Entire body is covered by a hard chitinous exoskeleton. Exoskeleton material important, chitin. Chitinous exoskeleton. What is chitin structurally? Huh? N-glycosamine. Okay. Is it a carbohydrate? Chitin? It is a polysaccharide. Homopolysaccharide, heteropolysaccharide. Glycosamine is a heteropolysaccharide. Okay. Brown in color. In each segment, exoskeleton has hardened plates called sclerites. Hardened plates called sclerites. Tergites dorsally and sternites ventrally. Okay. This is important. Terga and sternum. Terga dorsally. Dorsum is what? Dorsum is underbelly or back? Dorsum. Dorsum always is that which faces the sky. Ventral surface is that which faces the ground, earth. If you are walking on your legs, if cockroach is walking, the underbelly is the ventral surface, the back is the dorsal surface. So where do you have tergites? Tergites are present on the upper body. Dorsally, sternites or sterna are present on the underbelly, ventrally. Which one is harder? Dorsum or ventral surface? Dorsum is harder. Okay. That are joined to each other by a thin and flexible articulate membrane. arthrodial membrane. Head is triangular in shape and lies anteriorly at right angles to the longitudinal body axis. It is formed by the fusion of 6 segments and show great mobility in all directions due to flexible neck. The head capsule bears a pair of compound eyes. You see the diagram there. Compound eyes, that is the head capsule. Bears a pair of compound eyes. A pair of thread-like antennae arise from membranous sockets lying in front of the eyes. Antennae have sensory receptors that help in monitoring the environment. These are all not complex things, easier things. Anterior end of the head bears appendages forming biting and chewing type of mouth parts. What do you mean by appendages? Appendages are attachments. Attachments. They are detachable parts. They are not part of the skeleton of the head. The mouth part consists of labrum, upper lip, a pair of mandibles, a pair of maxilla and a labium. You can use that for labrum, and labium is a common reference to upper lip and lower lip. You can call it for humans also, labrum and labium. Upper lip is labrum and lower lip is labium. Mandible, maxilla. Mandible, maxilla. Pair of mandible and pair of maxilla, 2. We have how many mandibles? We have 1 mandible, 2 maxilla. This 1 mandible also, developmentally, there were 2 parts fused at the midline. All right. So 6 parts there. Labrum, labium, mandible, maxilla. A median flexible lobe acting as tongue. A median flexible lobe acting as tongue, hypopharynx. Use analogy of your own mouth. Visualize your mouth becoming cockroach mouth. All right. Visualize your mouth becoming cockroach mouth. Antenna also. Okay. Lies within the cavity enclosed by the mouth parts. Thorax consists of 3 parts: prothorax, mesothorax, metathorax. The head is connected with thorax by a short extension of the prothorax known as the neck. Each thoracic segment bears a pair of walking legs. So legs are present in the thoracic segments. All 3 segments have a pair of walking legs, 6 legs. Arthropoda, it is arthropod insect, so it has 6 jointed legs. The first pair of wings arise from mesothorax and the second pair from metathorax. So each segment, if you look at, prothorax has what appendages attached to it? Prothorax has the first pair of legs. Mesothorax has a pair of legs and a pair of 4 wings. Metathorax has a pair of legs and hind wings, another pair of wings. 4 wings, mesothoracic, called tegmina, are opaque, dark and leathery and cover the hind wings when at rest. The hind wings are transparent, membranous and are used in flight. The abdomen in both males and females consists of 10 segments. In females, the 7th sternum is boat-shaped and together with the 8th and 9th sterna forms a brood or genital pouch whose anterior part contains female gonopore, spermatical pores and collateral glands. In males, genital pouch or chamber lies at the hind end of abdomen, bound dorsally by 9th and 10th terga and ventrally by the 9th sternum. It contains dorsal anus, ventral male genital pore and gone apophysis. Males bear a pair of short thread-like anal styles which are absent in females. In both sexes, the 10th segment bears a pair of jointed filamentous structure called anal cerci. Now this part is the one which will give you loads of questions. The abdominal segment, some 4 or 5 lines, gives you all confusion. Go slowly on that. Abdomen in both male and females consists of how many segments? The diagrams they have given is also not so good. We will recreate the diagram. So when you are making perspective notes, these are the things in which you can just do your own diagrams. There are 10 segments. 10 segments, let us say. 1, 2, we are not bothered about segment number 1 to 6. All activity is happening in segment number 7 to 10. We will mention only 7 to 10. 7, 8, 9, 10. Okay? 7, 8, 9, 10. We will list down all structures related to female on the left side, structures related to male on the right side. Alright? Now read again. Read again and start jotting down things. See, this is a skill that you have to develop. As you read complex things, you should be able to simplify it and represent it graphically. Your own diagrams, you don't have to copy these diagrams. In the females, 7th sternum is boat shaped. Sternum is ventral. Correct? We don't bother about that. Whether it is boat shaped or ship shaped or aeroplane shaped, no problem, let it be. Okay? What we are interested is, together with 8th and 9th sterna forms a brood or genital pouch. Brood or genital pouch whose anterior part contains female gonopore, spermatical pores and collateral glands. Alright? So if you look at it, something related to reproduction you can consider. Correct? If you look at the structures, the mentioning of the structures, female gonopore, genital pouch is genital brood or genital pouch, gonopore, spermatical pores and collateral glands. There are three things. There is a pouch, 7, 8, 9. What do we call this? Segment 7, 8, 9. Genital pouch or brood. Genital pouch or brood. Anterior part contains gonopore. I'll erase this. We will distort it, no problem. 7, 8, 9 has genital pouch. In the genital pouch, three things are being mentioned. What are the three things? Anterior part, gonopore. Whatever it is, whether it is right or wrong representation, don't bother. We want to just mention three things that are there in the genital pouch. Genital gonopore is there, then spermatical pores and collateral glands. Spermatical pores, collateral glands. How it looks in actual cockroach when we dissect it doesn't matter. We want to just represent that the 7, 8 and 9th segment has genital pouch with gonopore, spermatical pores and collateral glands. What they do, all of that, later story. We will not jump the ship and try to get everything associated with this at this point. Okay? We'll go slow. We'll not go anything beyond what is there in NCERT, and we will try to make sense of this once and for all. We'll complete. That's all. That's all is mentioned there. Then, males bear a pair of short thread-like anal styles. Anal styles which are absent in females. In both sexes, the 10th segment bears a pair of jointed filamentous structures called anal cerci. Now, this is the 10th segment. This is anal cerci, 10th segment. Anal style present only in males. Okay. Done? That's all? In males, genital pouch or chamber lies in the hind end of the abdomen, bound dorsally by 9th and 10th terga and ventrally by the 9th sternum. Now, how do we represent this? Ventrally it is 9th and dorsally it is 9th and 10th. Alright? Don't bother to go into so much detail. Right now, we will consider on the male side, On the male side, genital pouch is corresponding to 9th and 10th segment. 9th and 10th segment. That's all. So this entire paragraph which has been given, without looking at the book, this alone will help you recall. Both males and females abdomen has anus opening on the dorsal side. Both male and females have a pair of anal cerci, and only males have a pair of anal style. So if you have two pairs of tails at that end of the abdomen of the cockroach, you know it is male cockroach. If you have only one pair of those threads at the end of the cockroach, you know it is female cockroach. That's all. This much is clear? That's all. No, you just have to write this, and you will write genital pouch. This is gonopore. This entire thing is genital pouch, gonopore. This is spermathecal pore. STP, just not a standard abbreviation. Standard abbreviation STP means sewage treatment plant. Okay. And this is collateral glands. This is genital pouch of male. So you know this is female, this is male, anal cerci, anal style, over, done. This is one important structural representation of cockroach we need to know. Then we will go. We will look at anatomy and we will add on a little bit more. See, when we have this foundational knowledge, something else gets added to that. Something else more gets added to that. Alright, we'll go. Is this clear for everybody? Are you able to visualize? Tell me the head region. Okay. So far whatever we have learnt, we will recall. Head has triangle shape. Right angle to the thorax. Then antenna. All right. Compound eyes. Upper lip. Labrum. Lower lip. Labium. A pair of maxilla. A pair of mandible. Then tongue or hypopharynx. That's all. Mouth parts over. Done. Thorax. 3 segments. Prothorax, mesothorax, metathorax. Metathorax. Prethorax has a pair of legs. Mesothorax has a pair of legs. Metathorax has a pair of legs. Prothorax has no wings. Mesothorax has a pair of wings. Hard wings, leathery wings. Metathorax has transparent wings. Who covers whom? The tegmen, mesothoracic wing, covers the metathoracic wing. That's all. Thorax over. Abdomen, how many segments? 10 segments. All the reproductive structures in the segment from 7 to 10. 7, 8, 9. What is present in females? Genital pouch or brood. See, cockroaches, they are very, one of the reasons for their survival is this fellow, the collateral gland. The gland will provide material for producing that case. You see that cockroach egg, no? You have that black casing. These collateral glands will provide material for building that case. Spermathecal pouch or pore, pore will store sperms. It is not just a short window. Fertilize if you can or get lost. No. Wait, I will tell you when to fertilize. That kind of a situation in cockroach. Alright? Cockroach will produce a lot of eggs. Ovum. The sperms are stored in the cockroach, in the female cockroach. The sperms are stored in the female cockroach. Packaging of this sperm and egg, fertilized ovum and the sperm, fertilization happens. And it is packaged in that case, and the case is delivered in a warm, dark place. And these embryos will develop within that case and the baby cockroaches will come out. Alright. There is one, There is one counter that these pest control people give against using this sprays, hit and such sprays you use to kill cockroach. What they say is these sprays will put the female cockroach, all cockroaches under stress. While under stress, when the female cockroaches realize that the stress level or the threat is survival threat, they will quickly package these fertilized eggs. They will bring the eggs, they will bring the sperms, fertilize it and package it into cases and deposit the cases at multiple places. So, when you spray that, you are actually probably contributing to increasing the cockroach population. You are killing the adult, but the adult before it dies, it will make sure that it will keep all its wealth in a safe place where its future generations can come and use. See nature, why cockroaches are so great at survival. Alright? So, lot to learn from cockroaches. Though we don't like them to be in our homes, we should study them, their behavioral aspect, anatomy and all that, remembering their names, whatever structures are there. Not that. The behavior of cockroach and how they survive. The fight for survival has to be learned from cockroaches. They are the oldest, by far. Anybody else who has survived as a species for so long? Shark, huh? I don't know. Shark? I don't know. But I know for sure that cockroaches are at the leadership table of evolutionary survival. If you have a leadership board of people of species which has survived the evolutionary challenges for millions of years, cockroaches have a very prominent position in the cabinet of evolutionary survivors. All right. Whether cockroach becomes the prime minister or not, I don't know. We need to figure out. But cockroach is definitely in the CCS. CCS is Cabinet Committee on Security. That is comprised of Prime Minister, Finance Minister, Defense Minister and Home Minister. These four are the most powerful people in the country. Okay. South Block. South Block is the administrative block in Delhi where the bureaucracy of all these departments operate from. So South Block is the most powerful building in Delhi. Okay. So you get into bureaucracy, you become IAS officers, IPS officers, you will be aspiring to go to South Block. Anatomy. Now we will get into the body of cockroach, dive deeper into cockroach and understand how they are designed. The alimentary canal present in the body cavity is divided into three regions. Similar structure, foregut, midgut, hindgut. The mouth opens into a short tubular pharynx leading to a narrow tubular passage called esophagus. This in turn opens into a sac-like structure called crop used for storing of food. See, crop in cockroach is a structure similar to our stomach. That's all. All right. The crop is followed by gizzard or proventriculus. Gizzard or proventriculus. It is like our duodenum. It has an outer layer of thick circular muscles and thick inner cuticle forming 6 highly chitinous plate called teeth. So they have chitinous plates. When the squeezing of the gizzard happens, the food that is present in the crop of the stomach or at the gizzard level will get crushed and they will get broken down into pieces. It is for mechanical digestion. We have teeth to bite and send the food inside. They have put teeth inside so that food gets bitten when is chewed when it is inside. Okay. The entire foregut is lined by cuticle. A ring of 6 to 8 blind tubules called hepatic or gastric CK is present at the junction of foregut and midgut, which secrete the digestive juice. It corresponds to our digestive glands and probably the liver. The hepatic cercae is the liver of cockroach. Unlike human beings, where we have a very robust, well-formed, structurally superior gland, the liver, they have some tubular structures. They also function like glands. They are tubular glands, you can consider. They are tubes, blind tubes, That means on one side they are closed, on the other side towards the gut they open. So what they are made of, what kind of cells are there, how they function, it is more or less similar to our liver and digestive glands. At the junction of midgut and hindgut is present another ring of 100 to 150 yellow colored thin filamentous Malpighian tubules. Malpighian tubules are corresponding to what in our body? They are like our kidneys. They are like our kidneys. Malpighian tubules will remove excretory products from hemolymph. Cockroaches do not have blood. They have respiratory pigments? Do they have hemoglobin? No. It is hemolymph, common circulatory fluid, hemolymph. Malpighian tubules will help in the removal of toxins from the hemolymph. Alright? They help in removal of excretory products from hemolymph. The hindgut is broader than midgut and is differentiated into ileum, colon and rectum. The rectum opens out through anus. Alright? That's all. See, digestive system, if you see, there is correlation. The cockroach eats some food. Where does it go first? Into a short pharynx. Then it goes to the esophagus. Then it goes to the crop. Crop at its base has gizzard. Gizzard has chitinous plates. So in the crop and gizzard, there is squeezing of the food that happens. During that squeezing, mechanical squeezing, the food is being broken into pieces. As it goes down a little bit into the midgut, where at the junction of the foregut and the midgut, you have hepatic caecae, which will release all the digestive enzymes, Now the food starts getting digested, broken down, and as it goes down, it will encounter a ring of Malpighian tubules. Malpighian tubules is not associated with the digestive process. It is associated with the digestive tube. Our body has kidney positioned in a separate place, well-formed organ, but cockroach has not got kidney in a special place. You need to have these structures anchored to something, no? Liver is a separate organ, so it can be positioned in its own place. But these hepatic caecae are not organs, they are small blind tubes. So they have to be tied to something. They are tied to the gut and opens into the gut. Malpighian tubules, do they open into the gut? Malpighian tubules are not opening into gut, but they are tied to the gut at the junction of foregut and midgut. Now these Malpighian tubules are doing their function of excretion. Then beyond that you have ileum, cecum, all of this. Those segments of the intestine continues. What do? There is the digestion continues in those tubes also. By the time food mixed with the digestive juices reaches the end of the tube, absorption of whatever is digested will happen. Whatever is undigested is thrown out through the anus. Where is anus? In which segment of the abdomen? Anus is in the 10th segment of the abdomen. Towards the dorsum or towards the ventrum? Towards the dorsum. Anus is towards dorsal side. Okay. When we say towards the dorsal side, it is not on the dorsum, not on top of the body. Okay. If you draw a midline through the body of the cockroach, in the 10th segment, terminally anus is positioned dorsally. You understand? So it is like aeroplane only. You see the anus of the aeroplane? Alright. From the mouth through the body, if you draw a midline of the aeroplane, the midline goes and the anus of the aeroplane is slightly up. Correct? If you consider that as the 10th segment of the abdomen of cockroach, correct, actually aeroplane is like cockroach. So it is not on top. Dorsal side is not on top. Slightly above the midline in the 10th segment. That's all. Blood vascular system. Blood vascular, is this clear now? The body surface structure we have discussed, the segments we have discussed, anatomy of the digestive system we have seen. Blood vascular system also we'll see. Blood vascular system of cockroach is an open type. What is open type? Open vascular system. The fluid, circulatory fluid does not remain confined to the vessels. It is in continuity with the body fluid. Body fluid and the circulatory fluid are in continuity. That is why it is hemolymph. Blood vessels are poorly developed and open into spaces, hemocoel. Coelom is the body cavity which will have coelomic fluid. Heme or blood vessels will have blood, and because there is continuity between these two, the space in which the blood vessels open is the hemocoel. The circulatory fluid also opens into it, and the body fluid also is present there. Coelomic fluid also is present there. Visceral organs located in the hemocoel are bathed in blood, hemolymph. See, unlike our body, where you have to be fed nutrients through vessels, it's like if individuals are like different organs, if we are like different organs in human body, we have to drink milk through pipelines that reach us. But if we are all organs in cockroach's body, we are all swimming in milk. Correct? Increases survivability. You don't have to have specialized mechanisms of delivering nutrients. Nutrients are all around. Enjoy. Okay. That is what it means. The hemolymph, visceral organs. Visceral organs means soft tissue, important organs in the body, functionally important organs in the body. Visceral organs located in the hemocoel are bathed in blood, hemolymph. The hemolymph is composed of colorless plasma and hemocytes. Heart of cockroach consists of elongated muscular tube lying along mid-dorsal line of thorax and abdomen. What is mid-dorsal line? Along thorax and abdomen. What is mid-dorsal line? On the dorsal side of the midline. Again, you draw a line, assuming that the heart has to sleep on the midline. Assuming that let's say this is the midline. This is the midline. Heart has to sleep on the midline. There are two possibilities. You sleep on your back on top of the midline, or you hug the midline and sleep under the midline. What is heart doing? It is sleeping on the midline, resting on its back comfortably. It's not hugging the midline. So it is above the midline on the dorsal side. That is mid-dorsal. Mid-dorsal means that. So if you see that heart is continuous throughout from the thorax to abdomen. Thorax to abdomen you have multiple chambers. Small muscular chambers are there. So everywhere it is pumping. It is pumping blood. It is an elongated muscular tube lying along mid-dorsal and thorax and abdomen. It is differentiated into funnel-shaped chambers with ostia on either side. What is ostia? Ostia means hole. It's a pore. Blood from sinuses enter heart through ostia and is pumped anteriorly to sinuses again. What is sinus? Sinus is an open space. You have sinuses here. What sinuses are these? Mandibular sinus. You have sinuses here. You have frontal sinus in the frontal bone. These sinuses are all ventilated spaces. In our skull, these are ventilated spaces. There is proper ventilation happening. If there is blockage of the sinus opening, it leads to inflammation of the sinus. It is lined with very sensitive membrane on the inside. It leads to inflammation of the sinus. That is what will cause headache in many people. Sinusitis, inflammation of sinuses. All right. So heart, blood vascular system, you recall? What all comprises the blood vascular system of the cockroach? A mid-dorsal, elongated, muscular heart. Throughout the abdomen, thorax and abdomen it is present. Triangular pouches are there. Each of these segment has holes. and there are blood vessels around these segments, capillaries, opening into the body cavity. So you have heart, incompletely formed blood vessels, sinuses around this heart where there is pooling of hemocoel. There is pooling of hemocoel or hemolymph, call it whatever you want. Pumping happens from the sinuses into the ostia, through the ostia into the heart, and again to the anterior segments. It is just released. That's all. The fluid for circulation is hemolymph. Hemolymph, that is the circulatory fluid. You call this fluid hemolymph when it is within the heart or the blood vessels. But when it is outside of this in the body cavity, you call it hemocoel. Both the fluids are same. Both the fluids are the same. It is like when the fluid is inside the blood vessels, it is plasma. When the fluid is leaked out of the blood vessels and it is in some other blood vessel, it is in our body. It is lymph. When the lymph gets back into the heart again, then it becomes a part of blood again. So it is hemolymph and hemocoel are the same fluid. Hemolymph is the fluid that is in circulation within the blood vascular system of the cockroach. Hemocoel is the fluid that you find outside of the blood vascular system in the body cavity. The fluid is the same. One other major difference is the organs are not supplied with blood vessels. The organs are not supplied with vessels to deliver nutrients and essential substances to them. The organs are bathed in hemocoel. The organs are bathed in hemocoel. So all that you have to do is keep circulating the hemocoel and keep cleaning up the hemocoel. And along with this, you see, the gut is also passing along this. The heart is there in the mid-dorsal region. The blood vessels are there pumping blood in and out. You have digestive tube also passing parallel to this in the body. And attached to this digestive tube is the Malpighian tubules going around everywhere in the body, in the abdomen. Long Malpighian tubules. It is anchored to the gut, but it is spread out throughout the abdomen. So around it, wherever it is finding toxins, it is removing the toxins. So Malpighian tubules are cleaning up the hemolymph. Malpighian tubules are cleaning up the hemolymph. Heart and blood vessels are pumping and circulating the hemolymph throughout the body. So in this circulation, the chance of toxin containing hemolymph reaching the Malpighian tubule increases. You have to keep doing the rotation. Otherwise, whatever fluid is around the Malpighian tubules is getting cleared. It is getting cleaned up. But the fluid in other parts of the body doesn't get cleaned up. The heart and the blood vessels is ensuring that the fluid is in circulation within the body. Malpighian tubules are cleaning it up. Organs are all surrounded by this fluid. They take what they want, they throw what they don't want into the same fluid. Malpighian tubule cleans it up. Digestive tract picks up all the nutrients, pushes it into the surroundings. Again, it is also pushing it out into the hemolymph. Heart and blood vessels keep circulating. So simple. Primitive life form, but increases survivability. Okay. So we have touched upon the digestive system, blood vascular system, excretory system also we have discussed, but we will read it and again look at it. What else? Nervous system will be pending. Reproductive system will be pending. The structures we have seen. The specific functions of the reproductive system, we will look at it. All right? That's all. Okay? Cockroach.
What pump back the blood into the heart? What pump back the blood into the heart?
Sinuses pool the blood. Sinuses are in continuity with the ostia, the holes. When the heart is pumping, there is pressure gradient created for the blood to pump through the heart, into and outside of the heart. Okay. Right, all the best for tomorrow's test. So tomorrow we will have classes in the last two sessions. 6:50 to 8:30, 9:30 to 10:45, two sessions. Oh, this was not working all the way? There is no battery in this, I think. Okay, right. You can disperse. 12:30.
Sir, actually there are some sir and madam who wants to meet them for sure.
Okay.
Just have to be here, they talk to them.
From SST. Okay.